finally, after having discussed the skeletal muscle and all its properties and contractility, we will now see the properties of the smooth muscle. Let's begin. First point, there are two types of smooth muscle. Single unit smooth muscle and the other one is multi-unit smooth muscle. Single unit smooth muscle is lining the viscera. You know, all our viscera is lined by smooth muscle, and single unit means all these smooth muscle cells, they behave like a single unit. This is just a diagrammatic representation showing the smooth muscle cells, and they will all perform as a single unit. So, important thing, where is it present? The viscera. Viscera like GIT and other places. Multi-unit smooth muscle is where the smooth muscle cells, they operate as multiple units and not like a single functional unit. So where do you find the multi-unit smooth muscle? Iris, then arrector pili muscle, the muscle that is responsible for goosebumps. Then the blood vessels also have this type of smooth muscle. So these are the two types of smooth muscle. Second point is the electrical activity of the smooth muscle. You know, most of the things that we will discuss here will be in comparison with the skeletal muscle or cardiac muscle. We will compare smooth muscle with those muscle types. Now let's look at the electrical activity. You remember what was the RMP of the skeletal muscle? Minus 90 millivolts. RMP of a smooth muscle is less negative. So it's in the range of minus 35 to minus 45 millivolts. And though we are calling it as RMP, resting membrane potential, at most places the membrane potential is oscillatory. It's not resting at a certain value, but it is oscillatory. So what happens is, for example, in the case of GIT, the GI smooth muscle shows slow waves like this, which reaching the threshold, and at the top of the slow wave there is spike. So this goes on happening with a certain frequency which is called as basal electrical rhythm, which we will discuss in the GIT. But that is the electrical activity, that's how it happens. So you can note that the action potential in the case of nerve or muscle was different, and here it is in this manner, this for the smooth muscle. The next point, the sarcomere, I mean ratio of thin to thick filaments. Remember, in the skeletal muscle, the ratio of thin to thick filaments is 2:1. In the smooth muscle, that's not the case. In fact, there was an MCQ like this. In the smooth muscle, what is the ratio of thin to thick filaments? And the options were 2:1, 1:1, 1:2, or 15:1. You want to answer that just now? Well, the answer is 15:1. How? Let's see this. First of all, in the smooth muscle, there are no Z lines. The Z discs or Z lines which are present in skeletal muscle at regular distances in the sarcomere, that's not the case in smooth muscle. They are replaced with dense bodies. Dense bodies. So in the place of Z lines, there will be these dense bodies at regular distances in the place of those zigzag lines, Z lines. And arising on either side of these dense bodies, there are thin filaments. So the thin filaments arise on either side. Look, even on the other side of the dense body there are thin filaments, but I'm not showing it right now. Just first concentrate on this part. And among those thin filaments there is a thick filament. This is the structure of sarcomere in the case of smooth muscle. So you can see that the ratio of thin to thick filaments in the case of smooth muscle, it is 15:1. And of course, as I mentioned just now, it will be on either side of the dense bodies. So this is how it works for smooth muscle. The next point, in the case of smooth muscle, there is no troponin C. If you remember skeletal muscle, on the thin filament there is troponin C, and when the calcium is released to initiate the contraction process, that calcium combines with troponin C in the skeletal muscle. Here there is no troponin C. Calcium, when it is going to initiate the contraction, calcium combines with calmodulin in the place of troponin C, and one calmodulin binds 4 calcium ions. And when this combination happens, when calcium combines with calmodulin, there is activation of an enzyme called as MLCK, myosin light chain kinase. And then once the myosin chains are activated, then there will be actin myosin interaction resulting in contraction of the smooth muscle. It means activation of the thick filament is the necessary event for the contraction of smooth muscle, and therefore smooth muscle contraction is said to be thick filament driven or thick filament regulated contraction, because it was initiated only after myosin light chain kinase was activated. So it's in the thick filament. Why did I emphasize? Because skeletal muscle contraction is called as thin filament regulated. Look at the comparison. Skeletal muscle contraction is called as thin filament driven or thin filament regulated. And why is that? It's because the so-called active sites are present on the thin filaments. Those active sites are kept covered during the resting state of the muscle, and only when they are uncovered they can come in contact with the myosin heads and the contraction can be started. So it is the thin filament and the uncovering of active sites over thin filaments that is going to initiate the contraction. Therefore, skeletal muscle contraction is thin filament regulated contraction. The next feature is, and this is very important feature, that smooth muscle shows a phenomenon called as latch bridge. Latch bridge phenomena is typical of the smooth muscle contraction. Let me just tell you the background of this. In the skeletal muscle, we have discussed it that there is cross bridge cycling, and that determines how much tension will be generated, what will be the velocity of shortening, and so on. And that cross bridge cycling happens when the impulses are sent to the skeletal muscle. Look, skeletal muscle is not contracting all the time. Skeletal muscle contracts only when it is required to do so. So cross bridge cycling occurs only when muscle is required to contract. It's not contracting for 24 hours of the day, not continuously. Coming to the smooth muscle. Smooth muscle is required to contract very frequently, almost throughout the day in certain cases. For example, blood vessels or GIT or the viscera. So, if we need such a regular consistent contraction, then it has to be energy efficient. So what happens is, latch bridge phenomenon means after a certain percentage of cross bridge cycling has happened in the smooth muscle, then the cross bridges remain latched. Cross bridges remain latched like a door latch, and that means there will be no further cross bridge cycling. You remember we discussed this cross bridge cycling in skeletal muscle, where we've said that the myosin head will detach and then it will attach to the next active site. This was the cross bridge cycling. The myosin head and the active site in front of it is called as the cross bridge. So it goes on happening in the case of skeletal muscle, and then it generates the required tension. And then the muscle will relax, because we don't need the skeletal muscle to contract in a sustained manner forever, But smooth muscle, we need a sustained contraction for prolonged periods. And when the myosin head is detached from the actin or the thin filaments, it needs ATP. This whole contraction process requires ATPs. So, to conserve the ATPs, to conserve the energy, what happens is, after a certain cross bridge cycling has happened, means a certain tension is developed in the smooth muscle, then the cross bridges remain latched. It means what? It means no further cross bridge cycling. That means no further ATP consumption. So, therefore, because of this, smooth muscle produces a sustained tension. There is a sustained tension in the smooth muscle with lesser ATP consumption. Less ATPs will be consumed. So, smooth muscle can generate a sustained tension as will be required in blood vessel for vasoconstriction, or in the case of GIT or other places. There will be a sustained tension development with lesser ATP consumption and lesser shortening also. I mean, if the cross bridge cycling goes on happening, there will be that much shortening of the sarcomere, isn't it? But here, after a certain cross bridge cycling has happened, the cross bridges remain latched. There is no further cross bridge cycling, and therefore less ATPs are consumed and less shortening, and it generates a sustained tension. So that is a very critical difference between a smooth muscle contraction and a skeletal muscle contraction. And therefore this, in a nutshell, the characteristics of smooth muscle.